So this lecture is going to be focused on understanding the rest of the visual system. So in the first lecture we focused on the processing that happens in the eye, and now we're going to be looking at things that happen past the eye as we take that light information from the eye and before we move on, I want to just kind of give an overview for how basically all sensory systems operate. Sensory pathways will generally involve 4 steps. So the first step will be sensory reception, and that's where it involves the use of sensory receptors that are located in the periphery that detect a stimulus. And in the case of the visual system, this is the photoreceptor cells, the cones and the rods. So light enters the eye and then is going to be entering and interacting in the photoreceptor cells. So that's the sensory reception part. The next step is sensory transduction. And that is the step in which the physical world that is including the relevant stimulus is being translated into something that leads to a neuronal membrane change, a potential change. And so in that case, that's the transducin pathway, where the light interacts with the opsins in the cones, rhodopsins and rods, causing that molecular change that leads to the change in cyclic GMP and in sodium. And so that is the sensory transduction portion of the visual system. Today, we're going to be talking about transmission, which is how action potentials are propagated from the periphery into the central nervous system, and then perception, how an organism becomes aware of the stimulus when the brain receives sensory information. And so we're going to be covering these aspects, these 2 aspects we already covered in the first lecture. Okay, so the visual system is made up of several sub-pathways. There's one main pathway that we're going to focus on today. There are a couple of other smaller pathways, but still are very important, and we will be discussing those at later points. So the first part is the retina. We talked about that on Monday, or in the first lecture. The retina has ganglion cells that have axons that go into the optic nerve, right? So those ganglion cells run up to the optic chiasm. Chiasm is a word that means cross. It comes from the Greek word chi, which is kind of an X shape. So that's what we have here. It's a cross. Now, the axons from retinal ganglion cells in the eye make their way to the optic chiasm, and interestingly, only 60% of those axons decussate. They go to the other side of the brain. So in this case, the left eye going to the right side of the brain. 40% actually stay on the same side. So another way to phrase this is that 60% of the axons go to the contralateral side of the brain, the other side of the brain, whereas 40% stay on the ipsilateral side of the brain. So they stay in the left-hand side. Once they're past the optic chiasm, then those paths are referred to as the optic tract. So this is the optic nerve, and now we're talking about the optic tract because we're essentially in the brain. Nerves are outside of the brain. The main visual pathway that we're going to be talking about involves the ganglion cell axons terminating in what's called the lateral geniculate nucleus. So 40% of the ipsilateral ones ending on the ipsilateral lateral geniculate nucleus, 60% ending up on the contralateral lateral geniculate nucleus, or the LGN. Then from there, the LGN projects to the primary visual cortex, also known as striate cortex. So, and this is known as the optic radiation. So there's a big projection that comes from the LGN and then projects out into the visual cortex. There are other visual pathways, as I mentioned, that are also important. One is the pretectum. So there are projections to an area of the brain called the pretectum, and this is what allows the brain to control reflex control of the pupil and lens. So all those little minor adjustments to make the lens look close or far, or adjust the amount of light that's coming in through adjusting the iris and changing the size of the pupil. The pretectum is involved with that. We will not be discussing that. There is a projection from the eyes to the superior colliculus. It's a very interesting part of the brain. It helps with orientation of movements of the head and the eyes. And we will be talking about that in a couple of weeks when we talk about motor systems. So I'm not going to talk about that today. And then there's one last pathway that's important, which is the eyes project to areas of the brain in the hypothalamus for the regulation of circadian rhythms. I want to just briefly cover this because I want you to have an appreciation for the fact that there are different kinds of retinal ganglion cells. We only talked about two main types, on and off retinal ganglion cells, and there's lots of subcategories of those. But there's another kind of retinal ganglion cell which actually doesn't receive any input from the cones and the rods. So the retinal ganglion cells that we talked about are kind of depicted here in gray. These are the ones that have the cones and the rods, and horizontal cells play a role. And then you have the bipolar cells, there's amacrine cells. Those are the retinal ganglion cells that we will be mostly focusing about today, and what we talked about exclusively on the previous lecture. But there's another kind that I want to mention, which are called the intrinsically photosensitive retinal ganglion cells. So they do not receive input from rods and cones, and instead they have their own photopigment. So these are neurons, the retinal ganglion cells, that have their own photopigment, which is called melanopsin. And it is sensitive to blue light. And these intrinsically photosensitive retinal ganglion cells, when you shine blue light on these cells, they start to fire. And they have a very slow time course. So this is the change, electrophysiological change in a cone. Here's the light intensity here. You can see this is over minutes. And it takes minutes before these cells start to fire. So they're very, very slow compared to a cone, which is within milliseconds, right? So, and then will maintain its membrane change with that light intensity, whereas with these cells, they just start to fire a whole bunch. Anyway, these cells project to the hypothalamus, and they help us regulate sleep. And we will talk about them in more detail later in the semester, but since they are part of the visual system, I want to make sure that we cover that as well, because that's also testable material. So the visual field is split in the brain, all right? And I'm going to walk through this figure in some detail. So pretend that in front of you, let's say it's Christmas time, and you're looking at something that's red on the left side and green on the right side. So that is the visual field in front of you. You got red over here and green over here. Now, your eyes are not in the exact same position. There's a little bit of space between them. The right side of the visual field will be sending photons to your eyes, and they come at slightly different angles, right? Because your eyes are in different positions. But for both eyes, as long as the object is far away enough, that light on the right-hand side will end up on the left-hand side of each eye, right? So it's shown here in green. So the green light is ending up on the left-hand side of the eye. Remember, because the eyes create a reverse image on the back of the eye on the retina. And that means, of course, the red part that's in the left-hand side ends up in the right-hand side of each eye. Now, importantly, the eyes are not identical. They are mirrors of each other. So there's a nasal side and a temporal side to each eye. The nasal is the part of the eye that faces the nose, and the temporal side is the side that faces your temple on the outside of your head. So this green region projects to the left-hand portion of each eye, but because they are mirrors, the green region is on the temporal side of the left eye and the nasal side of the right eye. So functionally different parts of the eye, even though it's the same side, left-hand side. So if we continue to follow this green region, the temporal retinal ganglion cell axons, they go down to the optic chiasm, and those are the axons that stay ipsilateral. See, so it's the temporal axons that stay ipsilateral and project to the ipsilateral lateral geniculate nucleus. The nasal retinal ganglion cells, they actually cross. So those are the ones that decussate, and they cross the optic chiasm and end up on the contralateral lateral geniculate nucleus. And so what this means is that even though we have axons that are splitting and going from two different, the axons are from the eye are going to different regions of the brain. Some are going ipsilateral and some are going contralateral. The visual information, the green information, is ending up on the same side of the brain, but down slightly different pathways. So the left eye information is maintained separately from the right eye information, even though they are seeing the same green thing. And then the same is true for the red, right? So then the red of the left-hand side decussates and ends up on the right LGN, and then the temporal side stays on the ipsilateral side. Okay. So the visual field is split. And then from there, this LGN will project to the primary visual cortex, or striate cortex, and this is then all right visual field on the left-hand side of the brain, and then the opposite is true for the right-hand side of the brain. All right. Now we're going to talk about, so that's an overview of just the pathways. Now we're going to talk about the lateral geniculate nucleus of the thalamus. So the LGN is a major relay station of the visual system. And why do I call it a relay station? Well, that's because the thalamus kind of serves as the brain's sensory relay station. So let's talk a little bit of the thalamus. So in a coronal section, this is where the thalamus is located, right here. And it has somewhere between 30 and 40 subnuclei. So you can break it down into several different brain areas, and each serving as a critical relay station for the cortex and other brain areas. So the thalamus is organized so that each nucleus is responsible for specific functions. And we can break down these functions into 3 major subdivisions. Okay. So first is sensory relay stations. And there are different sensory relay stations that process auditory information, somatosensory information, but also visual information, such as the lateral geniculate nucleus. There are other modalities and subnuclei that process, that act as for motor relay, including the basal ganglia, as well as the motor cortex. We'll talk about those later when we talk about motor information. And then there are portions of the thalamus that are very important for processing emotionally relevant information. And we'll talk about that in the much later portion of the class. But today we're going to focus on the lateral geniculate nucleus. The thalamus is a very critical part of the brain. Alright, so the lateral geniculate nucleus of the thalamus is specialized to process visual information. We have the right and left lateral geniculate nucleus, and they receive input from the 2 optic tracts, which synapse here so that the third order neurons carry visual information onto the primary visual cortex. Okay? This is a close-up of a human lateral geniculate nucleus. The primate or human lateral geniculate nucleus is made up of 6 major layers, and fortunately, neuroscientists have decided to call them 1, 2, 3, 4, 5, 6, with 1 starting at the bottom and 6 out at the outer region. And this almost kind of looks like layers. It's almost like a volcano with the layers of like striations, like layers of lava that's kind of been poured on top of them. The central portion of the lateral geniculate nucleus, it represents the center part of the visual field generally, and then all leading to one side. So there are different layers of so these different layers actually now get more and more complicated. So layers 1 and 2 comprise the magnocellular layers of the lateral geniculate nucleus, and layers 3 through 6 comprise the parvocellular layers of the lateral geniculate nucleus. So these layers of the lateral geniculate nucleus actually have segregated inputs from the different eyes and the different visual fields. So this figure is complicated, and I don't expect you to be able to replicate the schematic from memory. There may be questions about some of these details, especially since the test will be open book. So we're going to walk through this a little bit in some detail so that you can have an appreciation for the complexity of how the visual system works and how different light information getting to the eyes is kept separate. So in this schematic, we have the left eye, we have the right eye. Here is the optic chiasm. The left visual field would be blue over here, and the right visual field would be red over here. That's not shown, but we can tell that's the case because the left side of each eye is red, and the right side is blue. So there's something blue over here, and there's red over here. Okay? So we've got red going down, staying ipsilateral on the left-hand side, and on the right eye, it decussates and goes to the left LGN. So this is a schematic of the LGN. And here we have the 6 layers. 1, 2, 3, 4, 5, 6. The left eye projects to layers 2, 3, and 5. The right eye, which is dark red here, projects to 1, 4, and 6. So the information, of course, it's the nervous system, so it can't be completely logical in order. You've got 1 and 2, and then 3, 4, 5, and 6 where they're interspersed. This is because layers 1 and 2 provide different kinds of light information to the brain than layers 3 through 6. These 2 pathways are known as, in green is the parvocellular pathway, and purple is the magnocellular pathway. The names magnocellular and parvocellular refer to the size of the cells in the LGN. So the magnocellular cells are relatively large. That's why it's called magno. So these cells are pretty big in layers 1 and 2. Whereas layers 3 through 6, they're relatively small, and they're known as parvocells. Now, the kinds of light information that the brain receives from these 2 pathways is actually pretty different. The magnocellular pathway that comes from 1 and 2 is largely responsible for movement. The light information there helps what regulates movement and to some degree contrast, but not color information. Even though there's some cone information that's received by layers 1 and 2, the kinds of retinal ganglion cells that come from the eyes to layers 1 and 2 are mostly sensitive to contrast, and they help with the organization of movement. Layers 3 through 6 are a separate pathway, and this is where a lot of the color information is regulated in the parvocellular pathway. The way the visual system processes color is really super interesting, but it's pretty complex, and it's beyond the scope of what we're going to cover in this class. So we're not really going to be talking too much about what's happening in the parvocellular pathway. If you want to learn more about it, I highly encourage you to read online. You can even go to Wikipedia, but certainly the textbooks associated with the course also provide nice detailed information about how this pathway works. It's really cool and interesting the way it's organized. But from here on, we're kind of going to be talking about what's happening within the magnocellular pathway. Okay. So to go over this once more, we have the visual system pathway, the visual field going to one side of the brain from the left eye and the right eye, ipsilateral and contralateral, but they're going to separate layers of the LGN. So the information from each eye is still segregated so that the brain can process the information from both eyes separately in order to provide things like binocular vision and that kind of thing. But then we also, within the LGN, have 2 outputs, which is the magnocellular pathway, which is really important for movement, contrast, and the parvocellular pathway, which is really important for color, as well as a bunch of other visual processes. But color, primarily, is the most important one. So the output of the LGN is to the visual cortex, and they project to the primary visual cortex, which is the first station of the cortical visual system. We refer to this, there's a few names in which the primary visual cortex is referred to. So primary visual cortex is one of the main ways it's referred to, Striate cortex is another name for the primary visual cortex. And then we briefly talked about, in the very first lecture in this class, about Brodmann's areas, and this is Brodmann's area 17. So it's also sometimes referred to as area 17, as shown in this figure here. I will be using primarily primary visual cortex or striate cortex. Just not going to refer to area 17. So this is showing what the visual cortex looks like, the primary visual cortex looks like, in two primates. The macaque monkey, which is a primate that it had been heavily used for studies on the visual cortex and how it regulates, how it controls visual perception. And then this is showing where it's located in humans. So if you're looking at the lateral side in humans, there's a little bit that you can see. But if you're looking at the mid-sagittal plane of the brain, the primary visual cortex goes along this fissure here, where you can see it kind of as this long stripe. In all mammals, the primary visual cortex is located here in the occipital lobe. Neurons in the cortex, the mammalian cortex, are organized in layers. You learned about this in Neuro 1, so I'm going to briefly cover this here again, because it's a very important concept that you guys need to understand. All cortex has 6 layers. So layer 1 is largely cell-free. Then we have layers 2 and 3, which in some parts of the cortex you can't really tell the difference, but in other parts you can. Then you have layers 4, which is broken down into 3 subsections, A, B, and C. Then we have layers 5 and layer 6. Now that is the cellular portion. Well, layers 2 through 6 is where most of the neurons, almost all the neurons, are located within cortex. Striate cortex or visual cortex has the same overall organization. What we're looking at here is just a small section of cortex in a section, a brain section. So someone had sliced, I think this is human, so a human brain, sliced it on what's called a microtome to make very thin sections, sort of like almost like salami. And then these sections were then put onto glass slides, and then they were stained with something called NISL. And NISL actually stains all cells, but because of the way it stains, you can tell the difference between neurons and glia. And it shows up kind of as a purplish or bluish color. And so it shows the cell bodies of cells, and neurons are included in that. And you can see the different layers. So it's a really easy way of seeing where cells are located within neural tissue. It's commonly used, and we're going to be seeing NISL stain over and over and over again. And so you should know what NISL or crescent violet, it's another name for it, is called. So there are different neurons that reside in different layers. The major type of neuron that you find in the cortex is referred to as pyramidal neurons. And they are called that because their cell bodies kind of have a pyramid shape. You can kind of see it pretty well in this one. And you'll find pyramidal neurons in multiple layers. There also are spiny stellate neurons as well. But you're going to find these different kinds of neurons in different layers. The lateral geniculate nucleus projects to these layers of area 4 primarily. There's tiny little projections to other areas, but most of the input comes to layers 4A and 4C. And this is generally true for most areas of cortex that are receiving input from the thalamus. The projections go to layers 4. So layer 4 is the primary input for most areas of the cortex, particularly from the thalamus. Then, from there, in a given area of cortex, you're going to find, and we're talking about a relatively small area, you're going to have these columns where the neurons then project to different layers. So there's interlayer communication, interlaminar communication within a section of cortex, with the outer surface here, the inner surface here, and you're going to find neurons going up and down across those layers. So there's inputs coming to here, and then they project in and out. Then there is output. So there's input to layer 4. Then there's processing that happens within the cortex. These are referred to as cortical columns. And then there's output. So in the visual system, we have some processing that happens within that part of the cortex. And then these layers, layers 2 and layers 4b, the neurons there, they project to other visual cortex areas and other areas as well. Layers 5 actually send projections to the superior colliculus, and then layer 6 actually sends reciprocal projections back to LGN. So it turns out that there's an additional connection back to LGN. So we'll talk about this projection when we talk about the superior colliculus. So that's going to be a little bit later. We're actually not going to talk about this at all, but now we're going to sort of refer to some of the, well, actually in a few slides we will be talking about what is happening from the output of primary visual cortex. First we got to talk about what's happening within primary. Before I go on, and so this is showing exactly that. So we're showing layer 4 here that receives the input from the LGN, and what you can see is that there's right, then left, then right, and then left, and right, and then left, and that you have this interspersing of the eye. So the eye information, even though it's seeing the same part of the visual field, we're still maintaining segregation between the eyes. So this illustrates the data that they had seen in this experiment, the scientists who worked on this. They presented bars of different orientations. So here's one that is horizontal, and then they rotated it more and more and more, and then they had vertical, and they rotated it more and more and more, back to horizontal. So they can just rotate over and over and over again. And they find the particular part of the visual field that the neuron is sensitive to. So it might be the upper right or the upper left or lower right. It kind of depends on, so if they're looking at the left cortex, which part of the visual field would they be looking at then? Answer that question. That's a question for you guys. So they're recording in visual cortex. Where would they have to put the bar in the visual field? On the left or the right? I'm going to let you answer that. Okay. So looking in cortex, they put the bar in either the left or right, depending upon which part of the cortex they're looking at, which side. And then if they rotate it, they find that in some orientations, so they're presenting the stimulus right here between seconds 1 and 2. So nothing, then they present it, and then they turn it off to get a baseline, and then see what happens afterwards. If they present a horizontal bar, they don't see anything. No spikes. If they start to rotate it, now they start to see some spikes. And as it gets to the vertical side, now this neuron really, really likes vertical lines, and so it spikes a ton. And then there's a little bit more spiking here, and then as it rotates back to horizontal again, it doesn't spike anymore. This particular neuron that was being recorded here has an orientation selectivity for vertical bars. And you can graph what that looks like by graphing spike rate and then different orientations. So this might be 90 degrees, 0, negative 90, right? Actually, probably 90 here and negative 90 here. That would make more sense. But at negative 90 or 90 degrees, there's very little activity. But as you get closer to its preferred orientation, that's when you see peak activity. And then as you go back, rotating to the other way, where it essentially looks exactly the same as this, its orientation selectivity goes back down again. A huge majority of the neurons in visual cortex that have bar selectivity will have a selectivity to some orientation. They are not all vertical oriented, and they're not just like some vertical and some horizontal. You will find neurons that will have orientation selectivity to bars that are rotated like this, which means this would be the opposite. Or neurons that are oriented like this, that this is their preferred orientation, and then this would be the least preferred orientation. So you're going to find basically every single neuron will have an orientation selectivity for a particular bar, and then the perpendicular bar actually ends up leading to the least amount of sensitivity. Okay, and this is just illustrating the preferred orientation of one particular neuron in the cortex. The guys who discovered all of this are David Hubel and Torsten Wiesel. So Torsten Wiesel worked under David Hubel. They did all of these experiments in the 50s and 60s. They did a bunch of experiments describing how the visual system works, primary visual cortex, on and off selectivity. They also did a bunch of work on neuroplasticity, which was really critical for understanding how it works. And that's I talk about a lot of their work in my neuroplasticity senior seminar class. Here's some videos, links to YouTube videos for this one in particular shows them describing how they accidentally discovered bar selectivity, which is really kind of funny and serendipitous. Like they got the Nobel Prize for medicine for this discovery, and they kind of discovered it on accident, which is really hilarious. And then this is another video sort of getting into a little bit more in depth on some of the work that they did, as well as some other scientists who did some work on visual system. It's an older video. It's like probably 30 or 40 years old, but it's interesting. It's worth looking at. Okay, so how does the brain build orientation selectivity? So we're going to kind of build this up. We already have talked about center surround orientation, that neurons in the retinal ganglion cells will have this center surround orientation where the center is stimulatory and the surround is inhibitory, right? Different LGN neurons will tend to have orientation selectivity that is also center surround. So neuron number 1 here will have this orientation selectivity that's pretty similar to the neurons, the retinal ganglion cells that it's receiving inputs from. Okay, and then this neuron here in LGN will have this orientation selectivity. This neuron here will have a center surround orientation selectivity, and this neuron here will have this center surround orientation selectivity. But these different neurons have are maintaining a retinotopic map. So they are maintaining where things are located in space, in vertical and horizontal space. and they are organized in a way that maintains that map. So there is a map of the retinotectal. There's a map. There's a map of the visual field in the LGN. So this map, you can look at it from this neuron. It'll be in this part of the visual field. This neuron is in this part of the visual field. This neuron is in this part of the visual field. This neuron is seeing this part of the visual field. Okay. Neurons in the LGN start to converge on pyramidal neurons in the fourth layer, where they receive multiple inputs from different LGN neurons. And this is where bar selectivity now starts to emerge. So these neurons, they'll project to actually multiple different pyramidal neurons, but the way that they are organized is that any given pyramidal neuron will receive input from multiple LGN neurons that will now have an emergence of bar selectivity. So if you take these separate center surround orientations of these different LGN neurons and you let them add up onto a single pyramidal neuron in the cortex, now you start to see a bar emerge where there will be in visual space, in the same visual space, part of the visual field that this is seeing, instead of having these separate center surrounds on the separate LGN neurons, what we're seeing is the combination of them all adding up on one neuron. Now this particular neuron is driven by the activity of a bar, which is stimulating all of these center surrounds, right? So if you have a bar that is illuminating all of these centers and not the surround, that's when you start to see a bar orientation selectivity. So now we have in the visual cortex the ability to see edges. This is essentially edge detection. And we can have individual neurons that'll see lines with different orientations in space. With this kind of activity, you will find neurons in V1 that also have direction selectivity. So a large number of the neurons in V1 will have what's called direction selectivity. So there will be a particular orientation that they're sensitive to within a given receptive field, but they are sensitive to the direction of movement. So in this case, we have neurons that move in one direction. So if they're moving from left to right, this neuron, as it's going through the receptive field, this bar, as long as it's the right orientation, will stimulate activity in the receptive field here. So this particular neuron will fire if the bar travels across the receptive field in this direction. For the same neuron, if you're continuing to record from the same neuron and you move a bar across in the opposite direction, you might get a little bit of an onset of activity, but if it's moving in the opposite direction, actually activity is suppressed. So when this bar is moving right, that's what this neuron is particularly sensitive to, but as it moves left, that actually reduces the activity. So now, not only do we have the ability to see edges and contrast, we have neurons that are specialized to see which direction those neurons are moving. This appears to be the result of convergence from other V1 neurons that have a particular orientation selectivity, but then they are converging onto neurons, other V1 neurons, giving them the ability to integrate the order in which these neurons have been stimulated. This uses the principle of delay lines. We will learn a little bit more about delay lines when we talk about the auditory system, but in short, the way this works, and it's important for you to understand this, is that in this particular neuron, this neuron is somewhat farther away. So this neuron here, the left number 2, is farther away from number 3 than, say, this neuron or this neuron. And even though action potentials are very, very fast, if they are farther away, there is a difference in the time at which that they reach that neuron. Okay, so there's a little bit of a difference. So it'll take a little bit longer for the action potential to travel down a long axon than a shorter axon. Just a little bit. But that difference is enough to allow for when a bar hits number 2, action potential starts, and then as the bar is going and then hits this one, and then the action potential is just a little bit behind this one, because this is shorter, they should reach this neuron, the postsynaptic neuron, at the same time. So this one starts firing, action potentials go down here, and then this one starts firing, action potential goes down here, and then they both reach at the same time. Same thing for this one. Now, if the bar is going in the opposite direction, it's going to hit this neuron first, and this has the shortest axon, so this starts firing, and it reaches the neuron pretty quickly, and then this bar, then the bar hits this one, and then it takes even longer, so this is even more delayed, and then the bar hits this one, and it's even more delayed, and none of them are synchronized. So none of the action potentials reach the postsynaptic neuron in a synchronized way, and so then this neuron does not fire. So the direction of the bar and the movement that it's making is the integration of different V1 neurons that have different orientation that have the same orientation selectivity, and that are sensitive to slightly different parts within the receptive field, but they have delay lines that are built in that allow for the synchronous arrival of action potentials in order to cause the postsynaptic cell, number 3, to fire. Okay, that's delay line. Individual neurons in V1, they exhibit what's called orientation selectivity, as we talked about. Neurons that are near one another within a column of cortex, they show very similar orientation selectivity. So if you take an electrode and you start recording from layer 1, 2, 3, 4, 5, and 6, and then you look to see what's the orientation selectivity of all those different neurons in the different layers of cortex. They all have a very similar orientation preference. So this is the distance along the electrode track, right? So A is shorter, and then B is a little bit deeper, then deeper, 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 deeper. And they all have a very similar receptive field location. So this is the center of the receptive field. The center of the visual field. And then this is showing what is the receptive field of each of these neurons. And they all have a very similar receptive field location. It's kind of, in this case, to the upper right of the center of the visual field. And then if you look to see what is their overall orientation preference, all of these neurons have a very similar orientation preference. So these neurons in this particular orientation column have a vertical preference in this part of the visual field. So all the receptive fields are located here, and they all prefer the same orientation bar. This is what's known as an orientation column. So these columns are referred to as orientation columns. All right, what if we go the other direction? Now we take an electrode, and we're going to stay within the same layer. In this case, we just happen to be taking an electrode and pushing it through layer 2 and 3, but we're going across different columns. In this case, what we're seeing is that the receptive field position appears to be changing a little bit. So there's slightly different receptive field positions, but even more so is that you're seeing different orientation preferences, but it's not random. So A has this orientation preference here of preferring a horizontal bar, then B is slightly turned, and then C is now basically vertical, and then D is an orientation in the other direction. So there's an organization to the orientations that are preferred that goes across this set of tissue. So you have a little bit of change in overall receptive field preference, but a major change in the orientation of the bar that is preferred. And it happens to be changing in a particular pattern, where the bar seems to be spiraling in, going from, say, 0 degrees to 10 degrees to 20 degrees to 30 degrees. It uses, there's a pattern in which the bars, the pattern of the preference of the orientation of the bar go across cortex. Those are referred to, so we have ocular dominance columns as well as orientation columns. So when you go down inside tissue, you have layer 1, 2, 3, and then layer 4 that's receiving the input from the LGN. You have an orientation column. This particular column has a particular preference of an orientation. And then the one that's next to it has a slightly different orientation that it prefers. And the one next to it has a slightly different orientation, but it has a pattern in which it's rotating. You can see that it's going from less horizontal to more vertical to less vertical to horizontal, back again. And it does this spiral that does, where the bar is turning as you pass through a single ocular dominance column. Now, the different stripes that receive input from the contralateral eye or the ipsilateral eye, left or right, these are referred to as ocular dominance columns. Ocular refers to the eye, and this particular part of the cortex is dominated by input to one eye. So if you close, say, the ipsilateral eye, and then you leave the contralateral eye open, you will see orientation selectivity in this column at this preference for that eye, as long as you're recording from the right receptive field. Now, if you close the eye and then you open the ipsilateral eye, you will not see activity happening in this part. So it's eye dependent. The eyes are separate. But if you move over just a little bit to the next orientation column, sorry, not orientation column, to the next ocular dominance column, then this eye will be sensitive to a similar orientation, but it'll be the other eye that is driving activity in this particular orientation column. So we have organization of the different eyes, and that's referred to as ocular dominance columns. This is dominated by the contralateral eye, and this is dominated by the ipsilateral eye, right? C over I and I over C. C is dominating I. And then we have orientation columns where they have a particular orientation that they prefer. And this is because we have the preservation of eye information being kept separate in the primary visual cortex. All right, so that's V1. And that's really the only part of the visual system in the cortex that we have talked about so far. Now we're going to expand to other parts of the visual system. So beyond V1, there are more than 24 distinct, what we refer to as extrastriate areas of cortex. So these areas no longer have very strong eye segregation. Information between the eyes starts to be integrated and processed together to give us, say, binocular information, more motion sensitivity, color, shapes, objects, and even face recognition. As we go up the visual field, we start to hit other visual areas. So V2 receives a ton of input from V1, and V2 will project to V3 and V3A, but also V3 receives input from visual area 1. Then we have V4, and then there are additional areas. One is called MT, also known as V5, which is really critical for motion. And then we have other areas that are involved with face and object recognition. Okay. When we get beyond the primary visual cortex, or striate cortex, we start to see that there's the emergence of two major streams of cortical visual processing that occurs. We break this down into the dorsal stream and the ventral stream. The dorsal stream is the dorsal portions of the visual cortex, and it goes dorsal in the brain, towards the top. The ventral stream goes ventral in the brain, and it moves its way in the occipital cortex and then into huge portions of the temporal cortex and the temporal lobe. These two pathways can be broken into two sort of kinds of processing that's done. In the dorsal stream, we have V1 connected to V2, goes to V6 and MT, as well as other areas. These areas appear to be particularly interested in motion and visual control of action. We refer to this as the where pathway. And that's because these neurons are particularly interested in motion and where things are located in the visual field. It also is it's heavily involved in control of the eyes and the arms to guide reaching. So we find neurons that will help with that guidance that are visually driven. Okay, so it's integrating that information. So it tells us where things are in space. That's what the dorsal stream seems to be most involved with. Then we have the ventral pathway. Again, it's V1 to V2, and then we have a connection to V4, which is kind of like the first major portion of the ventral stream, which then leads to inferior temporal cortex, and then this is referred to as the what pathway. So neurons in this pathway seem to be less concerned with where things are located in space, but more concerned with what you're actually looking at. So it seems to be heavily involved in form recognition, object representation, and related to memories. So let's talk about the where pathway first. So neurons in NT are particularly sensitive to movement. This is showing some data taken from doing functional magnetic resonance imaging, okay, or fMRI. So fMRI, essentially what it does is that it's a measurement of neural activity, a change in neural activity over time. And the time scale is a little bit long, it's quite a bit longer than what an active potential might be. But if a brain area is firing a whole lot, you will see a change in the readout of fMRI. And what that change is seeing is change in blood flow. So if an area is particularly active, it will have a little bit more blood flow, and you can see that in an fMRI. That's essentially what fMRI is looking at. So in this case, this is experiments that were done with, I believe, humans. And they're doing fMRI here. And humans were presented with dots. And so the dots, if they were moving, so if the dots are kind of moving away in different directions, neurons in the activity in NT went way, way high. So it was super high. But as soon as those dots stop moving, activity in NT goes way down. And this happens within a couple of seconds. NT, in particular, seems to be heavily involved with tracking where things are moving and determining and having a focus on how much is moving and which direction they're actually moving. And it helps with the processing for that. So the neurons in MT, in fact, you can do experiments where you have a whole bunch of dots and only a few of them are moving, and the subject has to tell you, oh, they're moving to the right or to the left. And the more dots that are moving, the more activity that you will see in MT. But you will see that the activity in NT will go up as soon as right before the person will acknowledge that the dots are actually, when they make a decision of which direction the dots are moving. So there's a lot of processing about which direction things are moving and if there's movement at all happening in MT. It's pretty cool. There's a ton of interesting work in MT. fMRI is a critical, non-invasive way for us to see how neurons act over a relatively long time course, seconds. And there has to be a lot of coordinated activity in order to see those changes. It's a hugely invaluable tool for a lot of cognitive neuroscience and allowing us to understand what's happening within humans. And so kinds of experiments that you can do in humans, you just simply can't do in animals because of difference in cognition. We use fMRI to do a lot of those experiments. But there are times in which we can get a little bit more fine-grained when we can look at the activity of spikes. And we've seen a lot of what that looks like. One issue that arises, though, is that when you present something, you offer some kind of stimulus, like in this case we're some stimulus is being delivered, like let's say there's a light that's off, and then all of a sudden the light goes on, and we're recording from a single neuron. And we do this 20 times, so 20 different trials. This neuron apparently, let's just pretend that light is being turned on at time 0. This neuron has a certain spontaneous firing rate that's relatively low. You can see that, like in this trial 1, it only fired once. Trial 2, it fired 3 times. Trial 3, it fired 2 times. There's no real coordinated pattern though. This is at random. As soon as you turn the light on, now the spike rate goes up. Now this neuron is firing over a period of 500 milliseconds like 10 times instead of 1. And then in trial 2, instead of firing 3 times in the previous 500 milliseconds, it fires like 8 times. There's obviously, if you look at the sum total here, there's a lot more activity post-stimulus presentation than pre-stimulus presentation. But if you were to look at any one particular what's called raster, you would see, you might think that, okay, well, this is the pattern, that there must be a pattern here. But if you do it over and over and over again, you'll see that there isn't really a pattern necessarily. Sometimes you can see a pattern in a raster plot, but what it can be really useful for is just seeing that overall, over time, there is this kind of increased level of activity relative to this. And then we can create a histogram of seeing the of how many spikes normally happen within the first 500 milliseconds post, and then second half of the of 500 milliseconds as well, and then comparing it to the 500 milliseconds before, and you'll see that the spike rate would be low, and then it'll go up, and then it'll be relatively high. So that's how you read a raster plot. You just look across the different trials, and it shows you clearly something that's being presented at time 0, whatever this blue line is causing this neuron to increase its firing rate. Okay. So that's how you look at a raster plot. When you go up the the ventral visual system, you find neurons become more and more specialized. And we're going to show a little bit of what's happening here. So if you look in the on-center retinal ganglion cells, you have this center surround orientation. That is the typical stimulus that will drive an on-center retinal ganglion cell. This is also true if you look in the LGN. You're going to find mostly neurons that are have this center surround orientation. Now, when you look in V1, we get a little bit more complicated, and it's a little bit more specialized. So instead of center surround, we have bars that it's sensitive to. Then, as you get up into the temporal visual cortex, we find neurons that actually are particularly sensitive to faces. And then, even in some places, it appears that there appears to be specialization for identifying specific individuals that are important to that individual. So, such as the grandmother. And this is the idea of the grandmother cell. In your brain, you must have neurons that recognize that your grandmother is your grandmother, right? There has to be some place in your brain that is making that connection, and that you would see neurons that are increasing activity when that connection is being made. This is what a raster plot might look like with different presentations. So now we're looking at really a ton of different trials, right? And so each line is a different trial, and there's a bunch of little tiny dots. And you're doing it over and over and over again. And in this particular paper, what they did is they had one session, and then they did another session and another session, and they're separating them with different blue lines. They're looking for stability of how this particular neuron fires. And so this is one presentation. They present some stimulus, and you can see that there's kind of an increase in spike rate over time to this one, like right here at around 250 milliseconds post presentation. And you can, if you look at the average, you can see the number of spikes per second is somewhat consistent, and then there's an increase here. But for a different presentation stimulus at the same time, around 250 milliseconds post presentation, this neuron appears to be particularly sensitive to whatever this presentation is. So let's look a little bit more of what that is. So this data was taken from recordings of macaques. So these were macaques that were where they were doing chronic recordings of individual neurons over time. And each of these sessions is broken down by date. So they did May 21st, 22nd, 23rd, all the way out to June 7th. So this is a few weeks that they're doing this. And they presented these macaques with, and so they're recording in the ventral visual stream in the temporal cortex. They're presenting them with different faces. So they presented these macaques with the face of one of the researchers, and the macaques actually seem to have some kind of response to that particular face. But they also presented the macaques with faces of other macaques that they knew. So these are macaques taken from a colony, and so this would be Charlie and Frank and Bob. And whatever macaque that they were recording from, this neuron was sensitive to this particular face, as well as this face and this face. And so they could see a spike in activity. This was a macaque face sensitive neuron. And it was somewhat sensitive to the human face, but it wasn't sensitive to other faces of other animals, such as a lion. In fact, there appears to be a decrease. And then when you show objects like a violin or something else that's totally not a face, if anything, you can see that there's a suppression of activity. So these neurons are specific to faces. And not only are they specific to faces, they are specific to faces consistently over time, over a period of weeks. This one particular neuron sensitive to faces over weeks. So face selective neurons, they maintain constant visual response across not just weeks, but months. Okay, so in this study, this is actually a study that was done in humans. Some humans, when they're having neurosurgery done, they will volunteer, be able to be asked to, and they will volunteer to participate in studies. This is someone that was having, I believe, an electrode implanted into their hippocampus in order to help with seizures. But when you're implanting this electrode to help control the activity of the brain to try to prevent seizures, when you were doing it, you can also record activity. And in this study, they were recording in the hippocampus, and they were presenting these individuals with images of famous people. And one particular neuron that was seen here, they were showing different faces of individuals. When they showed this person the face of Halle Berry, this neuron fired. So that's shown here. So this is the raster plot across multiple presentations of Halle Berry. And during the presentation of Halle Berry, you can see that this neuron really liked it and fired pretty soon right after the presentation of Halle Berry, of this particular face. The same neuron responded to this face of Halle Berry, this face of Halle Berry, this face of Halle Berry. You can see that visually, they're quite a bit different. Like this one's brighter. Her face right here is overexposed. There's a very dark background here and here, but not so much here. What this neuron seems to be firing to is faces of Halle Berry. It also responded to a drawing of Halle Berry. So just completely black and white, totally devoid of color. Also responding to Halle Berry. It responded to a picture of Halle Berry dressed as Catwoman. So this neuron is also firing to Halle Berry pictured as a Catwoman, right? So it visually looks very different from this to this, but if you knew that Halle Berry starred in the terrible movie Catwoman, you would also have that response. But even more crazy is that this neuron also responded to the word Halle Berry. Clearly, the word Halle Berry has nothing, it does not look anything like Halle Berry, but it's the concept of Halle Berry that this neuron is firing to. So this is a visually sensitive neuron. You have to present some sort of visual stimulus to it, but it's conceptual more than just visual, than just the straight up visual information. This is a neuron that's a Halle Berry neuron. And then if you present other stimuli to this neuron that are not Halle Berry related, it actually doesn't respond. Interestingly, it didn't respond to Catwoman, but it definitely didn't respond to Bill Clinton, and Jennifer Lopez and Oprah Winfrey. So it had a little bit of response to Oprah Winfrey. In the same individual, they also found other neurons as well. I think that they, if I recall, they found neurons that were sensitive to Cameron Diaz. So, person's a big fan of movie stars from the 90s and early 2000s. All right. So here are some key questions about the visual system. How is visual field information organized in the brain? So knowing like which parts of the brain have different parts of the visual field and how we split that up. How is light information organized in the lateral geniculate nucleus? And what is the lateral geniculate nucleus connected to? And how is it connected? Using parvocellular and magnocellular information, right? What is the striate visual cortex? How do we know that it's called the striate visual cortex? What are ocular dominance columns? How were they discovered? What is orientation selectivity and what are orientation columns? And how were those discovered? What is the role of the dorsal visual stream and what is the role of the ventral visual stream? And then how does cell selectivity change as you move up the ventral stream? You have to know all of those concepts. And I look forward to talking about this at our next discussion section.